Hello everyone, welcome to the first class of circuit design. That in last class, we took you had a glance at what iGEM and what synthetic biology actually do. So this class will dig a little bit further in a circuit design perspective to show you what will you do as a circuit design member in iGEM and show you a little bit more knowledge of synthetic biology. After this class, you may build your general interest in circuit design and synthetic biology. So imagine that you are a computer engineer, that you don't just random connect wires together or random insert some codes to hope something will happen. Actually, you design a couple of logic gates like AND gates or OR gates and use some switches like on and off to perform specific task. So this is exactly what we do while we use DNA as our codes and wires, and our hardware would be a living cell to perform our test. You must have seen this picture, that this is the DBTL cycle of the iGEM. What we do is exactly as the design part of it. And after we build our genetic circuit, we deliver it to the wet lab for the test. Our workflow is very simple. First, we select a host cell, which is called chassis, which usually is a bacteria like E. coli or yeast. Second, we design a genetic circuit, which is a piece of DNA code that tells the cell how to do something new, like glow green when it sends some toxins. And third, we put that circuit into our pre-selected vehicle called a plasmid, so we can actually get it inside into the cell and assemble them, test them in the wet lab. Today, we're going to focus entirely on this delivery vehicle. If you pick a wrong plasmid, then your project will probably fail before it even starts. So select the right plasmid and know what does a plasmid do is vital before we begin all our works. In molecular biology, any vehicle that carries DNA into a cell is called a vector. Here are a few types of vectors that you might hear about. Here we have phage vectors, which derives from bacteriophage and use virus to inject DNA into the host cell. We also got BACs, which is bacterial artificial chromosomes, which can take massive cargoes and hold very huge genomes into the host cell. But for 99% of synthetic biology and in iGEM, we focus on plasmid. But for what reason? Because plasmids are the smallest and the easiest to handle, which to program them to do almost anything. A plasmid is just a circular loop of DNA that could live inside a bacteria, separate from bacteria's own main chromosome. Think that a chromosome is the bacteria's basic operating system, and a plasmid is some additional codes that we use a USB stick to plug to run a program that we designed. Another reason that we only select plasmid is that in iGEM, the limit of our genetic circuit is 30 kb. So even if we only use plasmid as our vehicle, it's already enough. Here is basic autonomy of plasmids or vectors in prokaryotic cells like E. coli and in eukaryotic cells. So we can see some shared elements like something called ORI, or you can see the origin of replication. Another is a selectable marker. On the top, you see a very similar but slightly different element. This is usually called a expression cassette. So in this cassette, you can see the same element called multi-cloning site. But upstream and downstream it, you can see some difference as the E. coli vector has a promoter and the Shine-the-Gallo sequence. And in the eukaryotic expression vector, we got Tata box. At the end of this cassette, you can see something called a transcription terminator or transcription termination sequence, which is basically the same. So here today, we will go through these fundamental elements in plasmids and show you some very important elements onto it. First, we will focus on this origin of replication, or you can simply say ORI. The origin of replication does two things. First, it decides who the plasmid can live. A origin of replication from E. coli usually won't work in yeast. The only way that they could possibly work is that it's a origin of replication designed universally. Second, it is crucial for design to determine the copy number of the origin of replication. Some origin of replication sequence is relaxed, which means they have some weak breaks. So these are high copy number plasmids, like the PUC families. They replicate very fast and in a large amount, making 500 to 700 copies inside a single cell. It's great if you want to harvest a large amount of DNA or produce a large amount of protein product. Other sequence, which have strong breaks, are low copy number plasmids. They may only generate 5 to 10 copies per cell. So you can use these plasmid if you're building a very complex circuit which cause a large burden to your cell, or you're producing protein which is toxic to the cell. If you make too much toxic protein, you will probably kill your host cell. And also another tip is that if you're trying to put two different plasmids into the same cell, they need to have some compatible origins, which means that they cannot in the same incompatibility group. If they use the origin of replication from the same incompatibility group, that the cell cannot recognize which plasmid is which, and when they do partition between different generations, you may probably get lost for one of your plasmid. Also, some other factors like the length of your insert or the strain you use, the growth condition, if it's optimal, and also the inoculum time will all affect your copy number. The second important element on your plasmid is called antibiotic resistant gene. So this is probably basically the security system that we try when we try to put the plasmid into our bacteria. Maybe you can only got one bacteria in 10,000 cells that actually intakes our recombinant plasmid. But how do we find that one cell? Is very simple, that we poison all. And we add the antibiotics into the food or the medium of our bacteria. The cells that didn't get the plasmid will die because we get our antibiotic resistant gene into our plasmid. The one that get our plasmid use this antibiotic resistance gene will survive. So there are some commonly used antibiotic resistant genes, like ampicillin is out here. It's very common, but it has a flaw that the resistant enzyme is actually secreted out of the cell and actually break breaks down the antibiotic in the surrounding area. So which means that the free non-resistant cells nearby, they may also survive, which is called some satellite colonies. Another bad thing about ampicillin is that ampicillin breaks down very fast, so you may have to use some fresh mediums. Another one is keramycin or chloride. Kanamycin or chloramphenicol, which are much tighter antibiotic resistance antibiotics. They pretty much kills everything that does not contain the plasmid of the cell. Uh, chloramphenicol is actually the standard of the iGEM shipping backbone, which is the PSB1C3. And also, if even you're designing for your keratic cells like yeast or even in mammalian cells, your plasmid usually still needs a bacterial resistant gene. So the whole plasmid could build on the bacteria first. Now we get into the fun part, which is the actual payload or where you put your cargo on in a classical way. So this is the expression cassette. So if you imagine your plasmid as a car and the origin of replication is the engine, the expression cassette is where actually the drivers put their cargoes on. So a classical expression cassette to make the plasmid a expression vector usually contains these few elements. First would be terminator or insulators in between the two end of the cassette to isolate the cassette. Second, the promoter could be seen as a start button, which recruits the RNA initiation factors to start reading DNA. The second one would be the RBS, the ribosome binding site. This could differ in which prokaryotic cells and also eukaryotic vectors. In bacteria, they use Shine-Darago sequence. And in eukaryotic cells, which is usually a Kozak sequence, or you can call it a Tata box. If you use the wrong RBS, the gene will be hardly transcribed into RNA. And then you got your gene of interest, GOI, or you can call it the coding sequence, CDS. This is actually where you put your insert gene in, and it's usually in the multiple cloning site, MCS. And it contains multiple restriction site for you to insert your gene of interest in. And eventually there's a terminator, terminates your gene. Here is an example of the plasmid P-SIVA441, and it's relatively simple. It's expression cassette, except for the multiple cloning site with multiple digestion sites. There is only one and two terminators around it without additional promoters and RBS. So this means that when you insert your gene of interest, that you may contain your own promoter and RBS region for it to actually perform the transcription. However, instead of the classical in addition to the classical way of assemble your gene of insert into the plasmid, some novel techniques like Golden Gate assembly or Gibson assembly allows you to basically insert your gene of interest into any site of the plasmid. So you don't necessarily need a expression vector or plasmid that contains this such expression cassette. We will talk about how to make a workable plasmid a little bit later. So, where do we get all of these plasmid resources? Well, we don't have to write DNA code from scratch every time. We have the standard for general science, like we use @gene as a database. We can search for the vectors that we want and order from such platform, or just simply get the sequence from it. Another usually used database is the SnapGene database. We can have the visualization map, and also a very great platform for visualizing maps to see exactly where restriction enzymes were cut. And also one last database that you can use is the iGEM registry for standard biological parts. So we have standard backbones there, which you can look up the sequence, see how the previous teams use it, and even see if it fails for them, that you don't make the same mistake again. So to wrap up, how do you actually pick up the right plasmid for your project? Next week, we would talk about how to practically obtaining them. And today, let's talk about strategy, that we have a checklist for you. First is about host compatibility. So that's the plasmid has a origin of replication work in your chassis. Make sure it matches. And the second is copy number. Ask yourself that, do you need a lot of the transcription expression product? If you need to purify a protein, you can go high copy number. But if you're building a sensitive genetic switch that could go breaks easily, go low copy number. Another thing is that for the selection marker. So if you're using two plasmids, they must have different antibiotic resistance genes. And also, if you're using two, make sure that their origin of replication isn't in the incompatibility group. The fourth is that, do you need a special function for your plasmid? That if you want to use functions like blue light screening, that you probably need a PUC plasmid. And also, like the lethal selection, it requires the deletion of the killer gene. And the last one is the assembly method, that are using BioBricks, Golden Gate, Gibson, or the traditional ligation technique for assembly of your gene of interest into your plasmid, to ensure that your plasmid has the right docking parts for your method, and to see if you need additional promoter, which would be like inducible or constitutive. Here are some other suggestions for your iGEM journey. You may take a look. Thank you.